Thanks a lot for coming, and it's a great pleasure to start this morning session on MDS biology and to invite our first speaker, Jan Stenerik from Sweden. Good luck.
Okay, thank you very much. I'd like to start by thanking the organizers for inviting me to give this talk. And what I will be talking about this morning, quite briefly, is about studies in my laboratory and in collaboration with others, trying to understand better the stem cells of myelodysplastic syndrome. And while the title might give promise that I'm going to provide any evidence on how one should target these cells, it's more going to provide insights into how some treatments are targeting or are not targeting these particular stem cells. And this is, and I have a disclosure I'd like to give. I always think it's very important when talking about stem cells, actually even in normal hematopoiesis, because those who are actually interested in normal hematopoiesis, even in mice, knows that there's still controversies about which cells that actually can act as a stem cell and give rise to all the other cells, and particularly which cells that have something we could call self-renewal. And that's very much also in the case of normal stem cells due to the limitations of assays we have. And I would argue that this limitation is much bigger when it comes to the issue of cancer stem cells, and in particular human cancer stem cells. And I think that's important to keep in mind, both with regard to the data I'm presenting and in general. But anyhow, this hypothesis that actually cancers in general and hematological malignancies in particular are driven by stem cells, whether they are rare or not, has been around for several decades, and it's really been driven by two historical observations. One was that one early observed in tumors that there's heterogeneity of cells in the clone, and the other is that one in different assays observed that only a very rare fraction could give rise to the tumor, whether it was in vitro or in vivo assays. And that raised two possible models: a stochastic model in which any cell in the tumor, or very many at least, could give rise to the tumor, and a hierarchical model very much colored by normal hematopoiesis, in which one proposed and John Dick, as I will summarize, later showed that only a rare population, a distinct population, had the key ability, namely to self-renew and to give rise to all the other tumor cells. And also very much, we should also keep in mind that in normal hematopoiesis, this is a unidirectional hierarchy, and that's also something one needs to keep in mind, that many of the studies assume that that's the case when they study cancer stem cells. So I just want to summarize some of the key historical evidence and also limitations. And it was really a breakthrough, and it remains a breakthrough, when John Dick and his laboratory demonstrated more than 2 decades ago by using immune-deficient mice that they could actually separate the population of cells, 34 positive, 38 negative, that fulfilled these requirements for leukemic stem cells. However, later work, particularly from the first author on one of these papers when she established her own group, Dominique Bonnet, demonstrated actually, and that still remains the case even with improved models, that many AMLs, you cannot get any cells at all to reconstitute mice. And that, of course, I do not believe that anyone believes that these patients would not have stem cells. So it really raises questions about negative data in particular. And in very important data, not primarily in AML only, but in general, basically demonstrated to us in mouse models that actually by making mice more and more immune deficient, and also in humans, but in mouse assays, that actually one could actually increase the sensitivity of the assays and actually get almost all cells in certain tumors to reconstitute the mice. And also I would want to emphasize that in very few cases people show a convincing hierarchical relationship between the so-called stem cells and others. So the ultimate goal, and that's we are probably, or some at least, are probably getting there, by, particularly by sequencing, is really to identify and fate map these cells. So it's not only about understanding whether they can act as stem cells, but also whether they act as stem cells in tumors. And this was actually, maybe surprising to many, very long time after John Dick's studies first convincingly demonstrated in pure mouse models just a few years ago. I will anyhow summarize some data from my laboratory quite quickly that we have published over the years that I would at least say gives a strong indication, particularly in low to intermediate risk MDS, of the identity of these stem cells and also the uniqueness of these cells. So Lars Nilsson in my laboratory demonstrated quite early on that both myeloid and lymphoid progenitor cells are involved in MDS. This was particularly shown in 5q minus MDS. And also, Peter Woll in my laboratory more recently demonstrated, and I'll show the key data, that there is a hierarchical relationship between these cells and other progenitors. And based on these findings, and showing that actually all the mutations that he could find in different patients in low to intermediate risk could be backtracked, that combined with the hierarchical relationship made us to make a few conclusions that I think are strongly supported, and that is that the MDS clone can only be propagated by these rare MDS stem cells, again, since we found no evidence of any patients in which we did not find the mutations back in the stem cells, and also that these recurrent driver mutations at this stage of MDS, not more progressed, seems to enhance the competitiveness over normal stem cells per definition, but they fail to confer extensive self-renewal because otherwise we would have found patients in which we did not find the mutations in the stem cells. In higher risk MDS, as in AML, I want to emphasize, and that's relevant for the last part of my talk with some new data, there is strong evidence that the self-renewal potential might be extended outside the stem cell compartment, and that and therefore that the stem cells might largely acquire a more progenitor identity, but nevertheless act as stem cells. So this is the key hierarchical data where Peter purified stem cells, transplanted into mice, and just like I told you about AML, there's a number of patients where he could not get reconstitution, but where he could get reconstitution, he could show that these purified stem cells, and this is, I don't have time to go through the details, but these are basically just gatings with different antibodies showing that you can generate all the different type of progenitors from these stem cells, whereas if he did the same experiments with the progenitors, he could not produce this finding. And also when he isolated these cells, he could both molecular and functionally show that these indeed are distinct progenitors, distinct from the stem cells. And this is just showing very simply that when he screened a number of mutations for recurrent mutations, and then basically saw to what degree they could be backtracked to the stem cell compartment, as you can see here, every single mutation or genetic aberration he found could indeed be found in the stem cell compartment. But again, this is in low to intermediate risk patients. And in other studies, actually preceding the studies I just showed you, Ramin Tiranchi and Peter Woll and others demonstrated and most of you probably know that lenalidomide is a very efficient treatment in isolated 5q minus patient or 5q minus in general. And in many patients at that time, when Lars Nilsson and these others study this, these patients, these are examples of patients that were reported to be in complete remission. And this is the most spectacular patient, I would say, but all of those patients were nevertheless principally the same, namely that when you look in the progenitor stem cells, almost all the cells are clonally involved prior to treatment. After the treatment, the progenitors and the whole bone marrow, we find no evidence for the clone, whereas in the stem cell, still a very large fraction. Nevertheless, this is a reduced fraction, as you can see. This just shows 3 different patients. Again, the same thing, a reduction in the involvement in the stem cell compartment, but it's nevertheless a very persistent population, despite of the patient being in complete remission, whereas in the progenitors, it seems to be completely gone. So in this treatment, these stem cells, rare stem cells, seems to be resistant, and as you probably know, these patients are not cured by this treatment, although they go in, very many go into very long-term remissions, and actually there's also disease progression under this treatment. So what I want to spend the last few minutes on is just summarizing some new unpublished data where we have done similar studies in allogeneic stem cell transplantation of MDS, and this is the only curative treatment, but despite of that, there are very many relapses, and many of these occur after long-term complete remission, in some cases more than 10 years. And this is, of course, typical for many cancers in principle, late relapses. And it's thought, although I would say the definitive evidence is not there, but that's something one needs to explore more, that preemptive therapeutic intervention might improve the outcome, which is very dismal for relapses. But we have lacked reliable biomarkers for early prediction, and also importantly for what we have done, the relapses predominantly come from the original founder clones, allowing one to use those mutations to actually track the clone. And this has been done quite recently, showing an increased risk for disease progression if the mutation is present shortly after transplantation, but nevertheless, in more than 20%, in this study, in more than 20% of the mutation-negative cases, the patients nevertheless later relapsed, and in 30% of the cases where there was mutations, they did not relapse within the time of the study, at least. So at the Karolinska University Hospital, we have performed a retrospective study of 23 allotransplanted MDS patients that were transplanted in these years. 12 of these patients had relapsed in this time span, and 11 patients remained in complete remission as a control group more than 38 months after transplantation. And Marius Dumitru, who led these studies, he used a highly specific digital droplet PCR screening for oncogenic mutations that we had identified in the diagnostic sample. And the goal was to assess measurable residual disease in remission bone marrow samples that had been taken at different time points routinely and on indication at different time points. So again, I want to emphasize that this was a retrospective study, and at the moment, the Nordic group is performing a prospective study to follow up these studies. And we investigated then for the presence of these mutations in whole bone marrow and in purified stem and progenitor cells. This just shows the different mutations in the patients. We had some early relapses that we defined 9 months or earlier, some late relapses, and then these continuous complete remissions. And we also looked in the relapse samples, and in only one case, I think, did we find an additional mutation. It was always the founder clone that remained at relapse in the relapse cases. This just quickly summarizes, since I have limited time, the key findings. So the mutations detected in unfractionated bone marrow samples, which is what is routinely done, was in 7 out of the 12 cases preceding relapse, but only by a mean of 3 months. So 3 months earlier, we could actually find evidence for the clone prior to relapse. In contrast, when we purified stem and progenitor cells, as I showed you, and these are rare cells, we could, in 11 of the 12 cases, actually detect MRD positivity in the stem progenitor cells preceding the diagnosis of relapse by a median of 10 months, and in some cases by more than 2 years prior to relapse. And in all 11 cases, there was a substantially enhanced sensitivity for detection of MRD compared to using unfractionated bone marrow cells. And I'll just quickly show a couple of examples of this. This is just gating on different cell populations here, on the stem cells over here, on multipotent progenitors and granulocyte mono, different types of progenitors, and showing 3 patients here. This is actually a sample from a patient that relapsed 7 months later. So the diagnosis of relapse was made 7 months later, here 10 months, and in this case as much as 27 months. And as you see in all of these cases with very high sensitivity, we could not find any evidence, so very much agreeing with the clinical diagnosis that this patient was in complete remission, no evidence. Whereas in 2 cases in the stem cell compartment, and in later samples, I want to emphasize that the clonal involvement of this population is, as expected, I would say, even higher, still while the patient is in remission. And in this case, actually, the stem cells were not involved, and rather the MPP population. Maybe in agreement, these are, of course, typically more advanced cases of MDS rather than low to intermediate cases. But again, variable which the populations were, but in all cases, some populations showed a high clonal involvement of the mutations that we screened for. And this is just to summarize and show The arrowhead here shows the time of relapse, and this shows the earliest time point we could detect in the stem and progenitor cells, and typically we then had multiple time points that we could find this where it remained negative in the unfractionated bone marrow cells. And very importantly, this is of course purely correlative, and it's also a prospective study, so the key thing was actually to look also in the patients that remained in long-term stable remission more than 3 years after transplantation. And as you see here, looking in multiple samples, neither with one exception in the very earliest sample, in no samples, in very many samples in these patients, can we find any evidence for clonal involvement in any stem and progenitor cells. So there was a very distinct difference in the patients that remained in long-term remission and those that we knew had later relapsed. So based on that, I just want to summarize what I think we know about MDS stem cell at the moment. In low to intermediate risk, it really is the stem cell population, that the normal stem population that harvests this population. I mentioned that this is the only cell that we think can propagate the clone at that time. In more higher risk, this seems to be a more extended population and diverse population. And as I showed you, both with pharmaceutical treatment and following transplantations, these seem to escape otherwise efficient therapies. And again, while I didn't offer you any, although that's what we're very interested in, of course, identifying targets that could target these cells, I at least showed you data showing that it might be meaningful to follow these populations, because they are, in the end, key to target, we think, to basically give a curative potential. And even in the case of, for instance, lenalidomide, they are also key to target, not to avoid progression towards leukemia. So I want to end there and start by thanking the highlighted people here are those people who participated in the unpublished studies I showed you at the end for allogeneic transplantations, and I particularly want to mention the postdocs, Marius Dimitro, Magnus Tobiasson, Teresa Mortera Blanco, and my very close collaborators at the Karolinska Peter Wolle, not least Eva Hellström Lindberg, who have been key for these studies. And again, we are following up this with prospective studies to get maybe more unbiased data on this. And a lot of other people have contributed to these studies over the years. Thank you very much.
Any questions? Okay, I'll start. So you elegantly showed that stem cells, MDS stem cells, are propagating disease and also the cause of relapse. If you were to design an assay or a research study to identify how to target these cells, although they are rare and heterogeneous, how you would design such a study? What would be your thoughts?
How to target them rather than just follow them?
Yes, but how to study that, even because they are rare, they are heterogeneous?
Yeah, so I think it's pretty clear, and I don't think it's a very novel thought, so I don't mind sharing it. I think in reality, since these cells cannot, by FACS for instance, be separated from the normal cells, and I think they're most interesting at the early time points where they still, as you saw, they're very clonally involved, but still the majority of those cells are not clonally involved. So I think single cell analysis is the only way to go to identify targets. One thing we know about these cells, and we are exploring already, that's also no surprise, is that compared to the other cells, they are dominated by very quiescent cells. So that's probably not the whole key, but we think that one of the keys is, for many treatments, is to activate them. And that's not only for chemo, but maybe even for immune therapy to upregulate targets that are relevant for immune therapy, for instance. So I think, like many other things today, although I think sometimes we do single cell studies with not having to do them because it's fancy, but in this case I think since we cannot separate them from the rest, we can enrich them, as I showed you, but we cannot purify them. So the only way to get at the targets, probably, unless they're really novel in the sense that they would separate, and I don't think they would be probably that different, we need to do single cell analysis.
Yes.
Very nice presentation, Stein Erik. Did any of these patients get prophylactic donor lymphocyte infusions after transplantation? And
sorry, can you repeat that?
Did any of these patients receive prophylactic donor lymphocyte infusions after transplantation?
Yes, they did a few. I can't, yeah.
And could you use this technique to looking at these stem cells, the abnormal stem cells, used to monitor the efficacy of the donor lymphocyte infusions?
Yeah, of course. I think in general, I mean, that's a fantastic thing with availability of bone marrow, and these are, of course, taken routinely, and we can even, of course, take them more. We have a possibility, although, again, I think the relevant question is probably to find new ways to target them. But anything we try to target them, we of course now have the opportunity to see what the treatment does to those cells or does not do to those cells. So yes, I think that goes for any treatment. In reality, one has the possibility to see whether to what degree one. And again, I think it's also very important, even for normal stem cells, like I said, even in mouse, we can still not purify stem cells to 100%. So these treatments also are nevertheless, particularly bone marrow transplant, are nevertheless very efficient, and I think they are also helping us to purify them better. So the treatments are, it's interesting from a treatment point of view to follow these cells, but it's also a good way to actually, for efficient treatments at least, to really get at the real stem cells, which are probably just a fraction of this population we are otherwise looking at.
Okay.
Thank you very much for that talk. Quick question, a few of your mutations in the relapse patients are associated with familial forms of myeloid neoplasm. So my question would be twofold, I guess. Have you studied the donors? Do you know if the donors could be carrying those mutations? And have you discarded that this is not donor-related leukemia in the relapse and not rather than the original clone?
Well, they're not donor-related in the sense that it's the exact same mutation. We specifically track the mutations of the patients, so that they're not. But I think I think the evidence for that would be very speculative, that it would be donor-derived.
Right, familial will have the same mutation, right? So if you have a RUNX1, it would be the same.
Very few, if I remember correct, I looked at the data this morning, very few of these are sibling, actually. They're unrelated donors, actually. I think there's two sibling in there, if I remember correctly. But yes, in those cases, it could definitely be the case.
Okay, thank you very much. So the next speaker is Manja Wobus from Dresden in Germany.
Good morning. First of all, I would like to thank the organizers for selecting our abstract for presentation. And I'm happy to present you a potential new mechanism for targeting the bone marrow microenvironment in MDS. So these are my disclosures. The pointer is disappeared from here. Maybe. Okay, so well, I tried without any pointing. So TGF-beta superfamily ligands such as GDFs, BMPs, activins, and TGF-beta itself are known to be very highly expressed or located in the MDS bone marrow. That results in an overactivation of SMAD2/3. And in turn, the inhibition of late-stage erythropoiesis. And that can be observed in the MDS patient by an anemic state. Ligand traps, such as luspatercept and sotatercept, are known to bind these ligands and prevent binding of the ligands to their receptors. Therefore, inhibit the overactivation of SMAD2/3 and can restore, at least in part, the erythropoiesis. In a phase 2 and phase 3 clinical trial, it was shown that luspatercept, which is a GDF11 ligand trap of activin receptor 2B, had very nice effects on the transfusion response of low and intermediate risk MDS patients. Although MDS is known to be a clonal hematopoietic disease, the involvement of the bone marrow stromal microenvironment in disease initiation and progression are well accepted. One main component of the bone marrow microenvironment are mesenchymal stromal cells, MSCs, which can differentiate into osteoblasts. And they actively support the hematopoiesis by the secretion of different cytokines, chemokines. And one important one is S.E.W or CXCL12, and we and others could show that in the MDS bone marrow, the S.E.W levels are significantly decreased. And interestingly, the SMAD-dependent BMP signaling increased the S.E.W levels, which could enhance the homing and engraftment of transplanted HSPCs. So the question for us was whether luspatercept, which has a direct effect on late-stage erythropoiesis, could also influence the bone marrow microenvironment. And to investigate that question, we used MSCs from MDS patients and age-matched healthy controls. For co-cultures, we isolated CD34 positive HSPCs from healthy donors. Recombinant GDF11 was used to induce SMAD2/3 signaling. And we used RAP536, which is a homolog of luspatercept with an identical ligand binding domain. Here you can see the methodical design. So we cultured the MSCs with GDF11 for 1 week in the presence or absence of RAP536, and then either directly analyzed the phospho-2/3 expression or activation and SMAD4 expression by Western blot. We analyzed the S.E.W expression and secretion in the MSCs. And in the second part of experiments, we co-cultured CD34 HSPCs for 1 more week on top of the pre-treated stromal layers, and investigated afterwards the supernatant and the adherent fraction of HSPCs, their phenotype, the clonogenicity, and finally we validated our in vitro results in an in vivo embryonic zebrafish model. First, we could demonstrate that RAP5D6 is effective to inhibit the SMAD2/3 phosphorylation and SMAD4 expression in human MLCs. So GDF11 induced a higher phosphorylation of SMAD2/3 and a higher expression of SMAD4, and RAP5D6 could inhibit these two protein levels. Please note that at the basic state, we have a higher phosphorylation of SMAD2/3 in the MDS MLCs, as well as a higher expression of the co-SMAD4. The SDF1 levels in both healthy and MDS MLCs were significantly decreased by the addition of GDF11 to the MLC cultures. And these decreased SDF1 levels could be again elevated by addition of RAP5D6 to the cultures. And it could be shown both at the mRNA and at the protein level. So this might have a functional influence on HSPCs, which were co-cultured on the pre-treated stromal layers. And indeed, after co-culturing for 1 week, the HSPCs, we could determine different populations in the adherent and in the supernatant fraction. Whereas GDF11 pre-treatment of MSCs caused a significantly higher fraction of cells which stay in the supernatant. The addition of RAP5D6 to the MSC cultures caused a significantly higher fraction of adherent HSPCs on these pre-treated stromal layers. That means it increases the more immature HSPC fraction. So having in mind that these mechanisms might be mediated at least in part by SDF1, we analyzed which other molecules could be involved in this interaction of HSPCs with the stromal layers. For the left-hand side, you can see proved again these higher SDF1 levels in RAP5D6 pre-treated MSC co-cultures. And also the CXCR4 receptor, it's the receptor for SDF1 on HSPCs, is higher expressed on these adherent hematopoietic stem and progenitor cells. And these higher CXCR4 expression, and maybe it's a response to the higher SDF1 levels, we could detect a higher expression of integrin. In that case, we proved for integrin alpha V beta 3. The increase in the adherent fraction of HSPCs on the RAP5D6 pre-treated MSC layers was quantified in LTC-IC assays in the counting of the cobblestone forming areas. On top, you can quite nicely observe that these higher numbers of colony forming units in RAP5D6 pre-treated co-cultures. And by quantifying that, we could show that the number of colonies was significantly increased after 2, 3, and 4 weeks of co-culture. Also, the clonogenic potential of HSPCs, which were further cultured for 2 more weeks after co-culture on the pre-treated MSC layers, was influenced by the pre-treatment of the stromal layers. We could demonstrate a significantly decrease of erythroid colonies after GDF11 treatment, and an increase after RAP5D6 pre-treatment. Maybe that is not so surprising in confirming that besides direct effects of RAP5D6, it has also an indirect effect on the formation of the differentiation of erythroid colonies. But interestingly, also the myeloid colonies were positively influenced after RAP5D6 pre-treatment of the MSCs. And that resulted in an increase in total colonies under that conditions. Finally, we wanted to validate our in vitro findings in an in vivo model, and we used here the embryonic zebrafish. The HSPCs, which were previously co-cultured on the pre-treated MSC layers, were injected into the duct of Cuvier. So they go directly into the circulation. And then they were analyzed in the CHT region, which is the hematopoietic organ of the zebrafish. And we could observe that HSPCs derived from RAP536 pre-treated MSC layers could be detected at higher numbers in the zebrafish. They survived for a longer time, and they also showed a more active migration capacity. And the quantification in the CHT region showed a significantly lower number of HSPCs after co-culturing on GDF11 pre-treated MSCs, and a higher cell number after RAP536 or co-cultured MSC co-culturing. And these HSPCs can quite nicely tract in a transgenic zebrafish, which has a green vessels here. On top you see the HSPCs, which were derived from the GTF11 pre-treated MSC core cultures. They are cells and they are happy, and they can be detected in the zebrafish. But if you compare with those derived from RAP536 or pre-treated MSC core cultures, then you will observe that more cells accumulate in the CHT region, and they are even able to extravasate. So to sum up, we can demonstrate that RAP536 or luspatercept is able to influence also the mesenchymal stromal cells by blocking GDF11 or binding to the ligand, to the receptor. Therefore, inhibiting SMAD2/3 phosphorylation, decreasing co-SMAD4 expression in MSCs, and since SMAD4 has different binding sites in the SDF1 promoter, and binding to these binding sites inhibits the SDF1 transcription, blocking of that binding increases the SDF1 expression in MSCs. And that in turn has a positive effect on the HSPCs, their interaction with the MSCs, the HSPC phenotype, their clonogenic and migratory potential. So we think that luspatercept therefore has a positive effect on the normal hematopoiesis or the remaining normal hematopoiesis in MDS, and besides its direct effects on late-stage erythropoiesis, it might support that the normal HSPCs, which might be important for the clinical outcome of the patients. At the end, I would like to thank all the people who have contributed to that work. Uwe Platzbecker for sharing that fascinating project. Nandini Asokan did all the zebrafish work. I would like to thank all the collaborators, Celgene and Acceleron for research funding. And I would like to thank you for your attention, and I'm happy to take questions.
I think we have time for one question.
Okay, thank you. Congratulations. Very exciting data. Did you have any chance to look if these effects that you describe also have an effect on mutated MDS progenitor, stem and progenitor cells?
So I didn't get
So-
it.
so
In
you-
total
you
mutations in.
So you show
Yeah.
that you have this effect on normal hematopoiesis.
Yeah.
Did you have the chance to specifically look at the mutated HSPCs?
No, so far we established all the methods and try to investigate some mechanistics, but of course we are continuing by also checking distinct HSPCs in the co-cultures, and then of course those also from MDS patients with a defined mutation state.
Thank you.
Okay, thank you.
Our next speaker is Ulrich Steidl from New York. He's going to talk about stem cell origins of MDS to AML progression. Thank you.
Great. Great, thank you very much. First of all, I'd like to thank the organizers for this opportunity. These are my disclosures. All right, brief introduction. You're all aware of the important role that has emerged of so-called pre-leukemic stem cells in AML and MDS pathogenesis. Can you see the pointer? Okay. And we know now that there's more than the bulk population that we have to pay attention to, and also more than the leukemia initiating cells or leukemia stem cells, but that there are precursor states that are partially transformed, not fully transformed, not fully transplantable in transplantation assays, that we and others call pre-leukemic stem cells. And these pre-leukemic stem cells, initially established in AML, are important for a variety of reasons. They're an important source of subclonal diversification and what I would call pre-tumor heterogeneity. But they're also, and it's been clearly demonstrated in AML, an important source of relapse. So it's not the initially transformed clone, but in many patients, relapses come out of these partially transformed pre-leukemic stem cells. And of course, the situation has become even more complicated, and you've heard about this from Ben Ebert yesterday, with clonal hematopoiesis essentially telling us that some of these mutations that are detected in these pre-leukemic stem cells are in fact compatible with healthy aging. And so it's become even more difficult to say what is a normal stem cell and what is a stem cell that's on the path to malignant transformation. And I just want to say the concept is not super new in the sense that there has been good evidence and early evidence, including from us in mouse models, that these pre-leukemic stem cells exist. Klaus Nerloff and others have contributed to this. Then, of course, Sten Eirich's work, aberrant stem cells in MDS, our own work from Chris Park's labs and others have contributed to this. And then obviously all the brilliant work in AML and in human AML, Ravi Majedi, Liran Schlusch and John Dick, and also Ben Ebert, Tim Lay and others that have shown the important role of clonal hematopoiesis in this context. Now, what's emerging as one of the key questions is obviously what's going on at this level of pre-leukemic stem cells and what really determines whether such pre-leukemic stem cells actually go on to leukemic progression or are compatible with healthy aging. And so we and others use both systems, both the human and the mouse, to study this. And as said by Sten Eirich in the previous presentation, I mean, it's great to study stem cells in humans in a translational setting, but it's very important to acknowledge the severe limitations of the assay systems that we're using, including the sorting schemes, but also the limited ability to transplant those cells. And so I think there's still a very important role for mouse genetic models of this, for the simple fact you have access to pre-leukemic stem cells and you have much better and precise, albeit not perfect, functional and prospective identification methods. And we and others have used this in the past 10 years. And I think have learned a lot of important things about MDS and AML pathogenesis. However, I want to tell you a story about one of our most recent human studies, and we're really interested in understanding how MDS progresses to AML at the stem cell level. And again, you know, the current model of progression, and you've heard again from Ben yesterday about this, is that clonal hematopoiesis progresses to MDS and AML in a somewhat linear fashion. That's what bulk sequencing studies seem to have indicated in the past 10 years. However, there hadn't been any specific studies of this problem that really looked at the stem cell compartment, and that's what we set out to do. So we collaborated with Amit Verma's lab at Einstein and wanted to really specifically look at this question and at the highest possible resolution at the stem cell level. So with Amit, we looked at longitudinal bone marrow samples of patients with MDS who then later progressed to AML. And we used a scheme, you know, using markers that other labs and ourselves had identified for AML initially, work from Paresh Vyash's lab, Craig Jordan, and ourselves, and threw all these markers together to come up with a new scheme and try to identify these pre-MDS stem cells, MDS stem cells and MDS blasts, and then of the same patient, after they have progressed, the same thing to isolate pre-AML stem cells, AML stem cells, and AML blasts, and at a really deep targeted sequencing, so we wanted to have maximum sensitivity at the stem cell level. You know, I can't talk about the definition of the stem cells and our functional validation in the interest of time. I'd ask you to look at our recently published paper, but we were able to really, for the first time, prospectively isolate functionally defined pre-MDS stem cells, similar what had been done with slightly different markers for AML and pre-AML stem cells before, and so that's the markers we used. And then again, our strategy was to sort all these populations, including a germline control, and then subject them to deep targeted sequencing of the 600 most frequently mutated and functionally relevant genes in heme malignancies. And what I would like to point out that through an on average 700x deep sequencing, plus the fact, you know, how rare these cells are, you see the percentages here, we're getting a functional depth of, you know, sorry about that, about 500 to 500 million x deep if you were trying to accomplish the same thing with with the sequencing of unfractionated cells. So what I would argue is we reach previously unattainable depth and resolution at the stem cell level, and I want you to keep that in mind because I'm going to show you some things that are different than what has been seen in bulk sequencing studies. So, you know, naively, when we started this study, we were expecting that we would see more mutations. The later we look, fewer mutations in stem cells, and then more towards progression. And you know what Jahao, brilliant graduate student in the lab who recently graduated, found was actually the exact opposite, which was quite puzzling initially. So what you can see here is that, in fact, in both the AML and MDS stages, the stem cells have actually more mutations than the bulk. But then if you look more closely, you see that, yes, the stem cells have more mutations, but those mutations are at smaller clone sizes, smaller variant allele frequency or cancer cell fraction. And then if you think about it, and we thought about it, then it kind of made more sense with the idea that we have a mutationally very heterogeneous pool of stem cells, but then only a few subclones get selected for and actually move on to become a tumor and a transplantable tumor. So you can look at this with different tools. It's highly statistically significant, and I just want to point out we saw this in every single patient. We looked at, I'm showing you one example here, you can look at all the others that we studied in the paper. The second thing we did, we used clonal inference models. I don't want to spend a lot of time on this. Again, you can now look at this in different patients at the blast level and at the stem cell level. And the only point I want to make here is that you know, the blasts, again, look simpler. In the stem cells, you have many more subclones that come and go that are detectable, sometimes only at one stage, and never again, so it's just much more complex. But what this suggested to us, that there's a distinct clonal evolution of stem cells versus blasts, and it's not just strictly a linear divergent clonal evolution as had been previously assumed. But we knew in order to really study this, we have to do single cell sequencing. And so we had to come up with a new strategy, a manual strategy where we took genes, mutated genes that we had found in the sorted but population sequencing, and designed custom primers for every single patient, and then performed manual single-cell sequencing into 96 well plates for every individual patient, matched this with the targeted customized primers, ran this through a Fluidigm platform, then with barcoding and sequencing. And what I want to say here, so this is very involved, very labor-intensive, but it was necessary because this method gives us a recovery rate of about 80%. What do I mean by this? What I mean by this is if we sort 100 cells, single cells, we get sequencing and mutational readouts of about 80 of them, which is a fairly high recovery. And as you all know, the commercial platforms that are out there and that we all use, including our lab, for other questions, typically they work, it's single cell data, but they only give you a recovery of about, in the ballpark 5%, 10%, maybe 15%. So you have to go in with tens of thousands or hundreds of thousands of cells to get a readout of a few hundred or a few thousand in the end. And obviously, for the question that we have in mind here, that's not feasible because we want to say if we don't detect something, it's really not there versus we've just lost the cell somewhere along in the process. And that's why we had to develop this method with a very high recovery rate. So what did we find? We looked at mutations. In every patient, we picked mutations that we call shared because they were picked up both at the MDS as well as the AML stages based on the population sequencing. But we also picked MDS-specific genes that we found mutated only at the MDS stage, or then AML-specific genes at the AML stage. And the idea was just to get a subclonal representation that would really allow us to look at the subclonal dynamics in the process. So what did we find? So this is the single-cell data now. You see each row is a mutation. Each column is a single cell. And in this patient, let's start with the blasts. We see an EZH2 clonal hematopoiesis mutation that's in the majority of stem cells, in the blasts. And you also see this at the AML stage. You see some subclones, and then you see two additional mutations here, NTRK3 and DUS22, that seem to occur only at the blast. So according to the old model, you would say, okay, these are the classic progressor mutations that actually mediate the progression to AML. Now, when we looked at the stem cells, we made a surprising finding, and that was that these progressor mutations were not only present in the AML stem cells and pre-AML stem cells, which was somewhat expected, but they were actually detectable in this particular patient 24 months prior to AML progression and in these sorted pre-MDS stem cells and MDS stem cells. And so obviously that leads to a model that is different than just linear divergent, and what you would argue is that there's a very early branching here at the stem cell level, and that the MDS blasts and the AML blasts evolve rather independently and in parallel rather than in succession. So this is one patient. I'll show you another example right here. So in this patient, we have a TP53 mutation as the clonal hematopoiesis mutation. We have the subclonal mutations, and here an ATRX and ERK mutation that seem to newly occur, again, progressor mutations seemingly upon progression to AML in the blasts. And in this patient, we made exactly the same finding. Not only was this mutation in the AML stem cells, but also in the MDS stem cells. And what was striking here was that it wasn't even a rare subclone at the MDS stem cell level, but actually a fairly frequent, a dominant subclone at the level of MDS stem cells. So here in this case, it appears that the MDS blasts were derived from a smaller stem cell subclone, and then for some reason this large stem cell subclone didn't come through at the MDS at the level of the MDS diagnosis, but only later when the patient progressed to AML. And this patient, there was about 18 months between the MDS and the AML sample. And then a last example, again, you can look at all the patients that we looked at in the paper that we just published a couple of months ago. This was a patient with an NRAS mutation, again, at the blast level, only detectable at the AML stage, but also in the sorted cells, we could also already in the population sequencing see that NRAS mutation at a low variant allele frequency, and this was confirmed by single cell sequencing. So again, leading to a model of very early branching and then rather parallel progression. So I want to kind of get to a summary and discussion of this. So what we think is that what the data indicates is that we have unmutated stem cells, yes. They develop to clonal hematopoiesis, but that the clonal hematopoiesis itself is not as homogeneous as we may have assumed, and that there's a lot of subclones that arise within this clonal hematopoiesis population. And then one subclone of those stem cells at the pre-MDS, MDS stage then develops and gives rise to the MDS blasts. But rather than those MDS blasts or those MDS stem cells progressing to AML, what seems to happen is that the stem cells, other stem cells in this pool, keep mutating, keep diversifying, and that then a different subclone gets selected for and then gives rise to the AML blasts and at the time of the diagnosis of AML. So you can further simplify it and say, so the linear model doesn't seem to be entirely correct, but that the process is really dependent on a highly diverse pool of pre-MDS stem cells, and that these pre-MDS stem cells evolve in a parallel manner and can give rise to either the MDS blasts or different subsets of stem cells give rise to the later AML clone. And in the patients we looked at, these are small numbers still. We looked in the paper, we looked at 7, we now have 2 more, 9 patients, but still small numbers. You can now distinguish extremely early branching, a little bit later branching, but it always happens at the level of the stem cells and not later. And of course, these findings have implications as we think for current bulk cell-focused precision oncology approaches that look at these at the MDS bulk. You can argue, you will not see what's actually ultimately driving the progression of the disease. So we are rethinking this currently. And so I want to say, again, we have low case numbers. This was a very involved technique. We are now expanding this to other areas, so we're very interested in understanding what's going on in low-risk MDS, in non-progressing cases, but also in normal older stem cells. We do have to rethink, in our opinion, our current approaches that are bulk cell focused, and we're thinking about the clinical feasibility of stem cell sequencing in patients with MDS, just to really increase the sensitivity of our detection. But then I think really amongst the most interesting are the mechanisms that we have to think about. And because really the question is, if the mutations are already there, why do they not get selected for early versus late? Or what determines this process? And I think beyond cell intrinsic mechanisms, genetic or epigenetic, we have to think about cell extrinsic mechanisms of selection, like the microenvironment niche, immune surveillance, treatment, and others. And just to expand on this a little bit in the last, and this is my last slide. So we now have these pre-MDS stem cells, but then from here on, it's really not a linear progression of the stem cells and to AML, but these pre-MDS stem cells seem to be inherently heterogeneous. That's what our work has shown, and I would like to say that, of course, John Dix and Liran's work, brilliant work in de novo AML, has suggested a very similar mechanism in relapsed AML, where they could also find so-called progressor mutations early on and at the time of diagnosis of the de novo AML, if you just look close enough in the stem cells. And I think that really leads us to believe that we have to study the stem cell subclonal selection process much more than just look at the mutations, because we have to understand why do they move forward, why do subclones break through, and when and how, and what are the factors that determine this. And you know, this, I think, defines some of our current research goals. So I have to acknowledge the people here. All this work, highly collaborative, would not be possible without a lot of key collaborators. First and foremost, Amit Verma and also Aditi Shastri, who is here at this meeting at Einstein. Fantastic collaborators who made all this possible. Other collaborators at Einstein, core facilities, and also our outside collaborators. And with that, I'll stop, and I'd be happy to take any questions. Thank you very much.
Yes, please.
Hi, Steve Gore from Yale.
Hey, Steve.
Good morning, Uli. It's really beautiful work, and it makes me get onto a very old soapbox of mine, so everyone can groan. But I wonder if it isn't time now, given what you show, that these so-called leukemic, if you will, progressing cells are present at the beginning, to get rid of these terms about leukemia and acute leukemia and pre-this and pro-that and all this other stuff, which is based on really antiquated morphological parameters, and which cause our regulatory agencies and our funding agencies and our scientists to think very dichotomously and fund 20% blasts and not 10% blasts and all this other stupid stuff we've lived with all my career and before that, and really go to some, I think we need to get together a bunch of people to come up with a more biologically based descriptor of this syndrome, maybe based on clonal size, proliferation, and other drivers that you may find, given that these cells are present at the beginning, because ultimately, one's goal of therapy might be to prevent the outgrowth of a clone that will eventually phenotype itself as what we tend to call 20.5% blasts AML, but it's all kind of silly. I don't know what you think about that.
I agree. Obviously, I mean, studies like this and the studies that have emerged in the last 5 to 10 years, they raise many more questions than they actually answer. But I think what is, and we've our hands full trying to go after those questions. But I think what's very clear to me is, and I agree fully with you, that just looking at blasts, also in our clinical evaluation, follow-up, therapy evaluation, this is just simply not enough anymore. And we need to understand also in the context of clinical trials what's happening to these stem cell subclones. I think this is of critical importance. It's not just the niche and the immune system, that's also important, but we're throwing therapies at these patients and we have absolutely no idea what these therapies do at a subclonal level in the bone marrow on stem cells, if we just keep looking at blasts. And I agree with that. It's not like I can propose a new consensus that everybody here in the room would agree with. But I think we should as a community, I think, talk about this and see what could we maybe agree on, because it's very clear to me, that just looking at the MDS blasts is not sufficient and will not get us where we need to be in terms of to make a really transformative advances in this field. I agree with that.
Ulrich, one question for me. So in AML, chemotherapy accelerates the selection of these leukemic stem cells. In MDS, even without chemotherapy, you get the selection. So what do you think is the similarity between giving chemotherapy and the evolution that happens in MDS?
Yeah, so, I think you raise a very important point. I think in MDS, it's more complex and subtle in the early stages. And also, there's different types of therapy, obviously, that are applied in MDS, including just, what we think are just supporting care agents. And we neither know what they do at the level of stem cell subclones, but we also don't know what they may be doing in terms of, let's say, the stroma. And that may lead to indirect effects on also the stem cell subclonal dynamics and competition. And I have no answer really to this, but I do think it's a very, very important topic, and we need to study this, you know, in addition to all the genetics efforts and everything that are going on anyways, and I think that have taught us a lot and will keep teaching us a lot, but I think in addition to that, we need to pursue those questions.
Okay, thank you for a great talk. We will move on with Erika Exioglu.
Good morning. First, I would like to say thank you to the committee for allowing us to present our work here. I would also, I forgot to put the slide, but I don't have nothing to disclose. First, I would like to talk to you about inflammaging. Our group focuses a lot on the microenvironment of MDS, inflammatory microenvironment of MDS. And inflammaging itself is the process of aging and inflammation together that gives rise to a lot of the pathogenesis that we see in many diseases. And we have actively looking at it in the context of MDS. In particular, we have been focusing on the damp S189, which has been making splashes throughout, and it has been shown to be an important molecule driving a lot of the pathogenesis. Our work has been very active at looking at this, and we have published a lot already about on the subject. And what we have been looking most of it is how S189 both directly and indirectly can influence the pathogenesis of MDS. Indirectly through the activation of myeloid-derived suppressor cells, which also produce S189 and can directly kill hematopoietic stem progenitor cells through PD-1, PD-L1 ligation. But we have also shown that S189 directly can induce pyroptosis of HSPCs, which also contributes a lot to the pathogenesis of the disease. We have actually shown as well that we can, blocking this process itself, we have tried with many therapeutic agents. One of them that we have published, we have shown that if you block this, you can prevent a lot of the genomic instability and a lot of the processes that I'm going to explain today. Other groups, including Dr. Ibert's group and Raji Maker's group, have actually worked on this, confirmed the importance of S189 on this pathogenesis of MDS, and actually shown that it's an active participant on the development of this phenotype, including the genomic instability that we see in the disease. What we have been recently worked on using the S189 overexpressing mice is that through the aging process, when we wait long enough, we can actually see that the mice start to show metabolic dysfunction. For instance, like we see a lot of very visible weight gain, which actually translates into a lot of fat in the bone marrow microenvironment, which leads to smaller space for their hematopoietic cells to grow. This is due to the activation of a very specific gene called FTO, or fat and obesity associated gene, which we actually shown is upregulated in MDS. And FTO itself is the methylase of adenosines in RNA, and we have shown that in total RNA there's consistent demethylation that can be induced by adding recombinant human S189. If we either overexpress FTO with a lentiviral vector in primary healthy bone marrow cells, or add S189 or their combination, it seems to actually decrease colony formation, and we can restore some of that colony formation by knocking down FTO with a CRISPR vector. As I said, FTO is a demethylase. So one of the things that we started to look in after we found out that it's there is that FTO, when there's demethylation, it removes SRSF2, which we all know is very important in MDS pathogenesis, as one of those mutations seems to be like one of the important things for MDS. What is important also about splicing components like SRSF2 is that when you remove it, actually can lead to genomic instability. And in an intermediate step, which I'm going to talk about later, is the formation of RNA-DNA hybrids, which are a consequence of the formation of R-loop structures in the nucleus. So what we see is that either through the addition of S189 or in MDS bone marrow, we see that there's colocalization of FTO with SRSF2, And what we see is that most of the SRSF2 is now colocalizing outside of the nuclei, so it's being restricted. So what we found is that this is also linked to the activation of genomic instability induced by S100A9, and that also leads to double-stranded DNA demarcation through gamma H2X, which can be removed which can be reduced if you again knock down FTO with CRISPR. And again, you have the formation of RNA-DNA hybrids in the nucleus, but surprisingly to us, we also found those RNA-DNA hybrids also in the cytoplasm of the cells, where they can actually serve as dams. So if we can actually prevent, so these RNA-DNA hybrids, other groups also have shown that our loops are important in MDS pathogenesis. And what we show is that if you use RNase H1 overexpression, which is an enzyme that can actually remove our loop structures from the nucleus, you can actually restore some of the hematopoiesis back in MDS bone marrow specimens. Because RNA-DNA hybrids themselves are DAMPs, you can actually, it has two putative receptors that can understand it. One of them is NLRP3, which we have shown previously is very important, is upregulated in MDS through the formation of pyroptosis. And the other one is TLR9, which we have actually found to be overexpressed in MDS, especially in the surface of the cells. So that's all I'm going to talk about that for now, but there's a lot more information about that. But basically, what our working model is right now for that is that through the aging process, you get activation of S100A9 that leads to a lot of metabolic and ribosome stress changes that lead to the formation of RNA-DNA hybrids, first in the nucleus, that get inactivate. This actually happens through the inactivation of RAMBP2, which is a nuclear molecule, and I can show, if you have questions about that, I can show you that separately, and leads to the activation of innate immune receptors like TLR9 and NLRP3. But this leads to a very important question. So the next things that we want to understand is like, so is the FTOs that the methylase, and this localizes SRSF2, would there be specific locations that lead to this, or does it randomly happen? So there's several things of how this happens, and we are in the process, although we couldn't have it ready for this talk, we're doing M6A sequencing and DRIP sequencing to try to understand not only where the M6A happens, if it is in specific spots, but also if they're linked to the formation of RNA-DNA hybrids. But we have done actually splicing analysis on some of the sequencing data that we have performed, and that's what I'm going to show you today. So based on some of the sequencing that we did with hematopoietic stem cells, like CD34 cells, in healthy versus MDS bone marrow, we actually shown as well, see as well that there's increase in adipogenesis genes in MDS, as well as oxidative phosphorylation, which matches our current model of what we think is happening internally. But there's also two things that are really important: DNA repair, which marks genomic instability, and modulation of cell cycle, which is to G2M checkpoint. In the mice, similarly, there's those things are involved, but when we look at a lot of the signatures linked to MDS, we actually see the aging process very well, where you can see that when you have young wild-type mice, there's a certain signature where some of the things that are important for MDS are actually restricted, and those actually get turned on in old transgenic mice. However, when we compare wild-type mice that are old with young transgenic mice, they seem to actually share a very similar signature that is fairly intermediate of that, which actually would say that suggests that S100A9 itself is kind of an accelerant to the inflammaging process that goes on in all of us. Based on that, we try to look forward into this, and since splicing is something that is very important, and SRSF2 is actually modulated in here, we try to understand what's happening. So we took advantage of the fact that we have high-depth RNA sequencing data. And through the bioinformatics core, they used They used a procedure highlighted here in this paper. They did splicing analysis of our data to look for skip exons, alternative 5' splice sites, 3' splice sites, mutually exclusive exons, and retraining introns. And we actually have been finding some interesting things. So for instance, we have that alternative 5' sites, mixed mutually exclusive exons, and skip exons are actually increased when we look at comparisons between, for instance, a comparison made on a regular aging, which is young wild type to old wild type, compared to something that is a regular aging for transgenic mice, or when we compare the old transgenic and the old wild type mice. And there's actually for retaining introns, there's actually instead a decrease on that type of splicing changes. So this tells us that splicing is, even though there's not common, is not like you're having an increase of all of them together at the same time. For specific type of splicing changes, there is an increase when you have S189 in there. When we look at the enrichment in databases, for instance, with the Go Biological database, what we see, for instance, marked with the green arrows, is a change, an increase on genes that are linked to splicing changes. So whenever you have all of these, you actually not only is splicing modified, but a lot of the genes that are there, they're actually splicing genes that are modified themselves. So that whole procedure is probably being changed. It doesn't happen that much in this database for the wild-type mice aging, but it was interesting to see that even in normal aging, you have markers like myeloid leukocyte neutrophil degranulation, which are linked to myeloid skewing of the cells. When we look at Go molecular function, we again see a lot of the green splicing linked, that is linked to both aging and S189. So S189 seems to be important for that, for inducing changes in splicing. And again, we see mostly in the wild-type changes in myeloid cells. When we look at reactome, though, the things were a little bit changed, and we actually see more things that are cell cycle dependent, circadian rhythm changes. For instance, it has been shown that FTO plays a really important role in AML transformation, and it actually plays also a role linked to the clock genes, which are very important for that, and they actually came in here like BMAL1 and circadian clocks in general. When we look at the same databases for the human, we actually see more a signature that looks more the matched signatures that we saw on all the mice. So we see components of DNA repair, DNA damage, and we see DNA helicase activity, things that are linked to the formation of R loops. We also see things like RNA binding. So there seems to be a lot of match to what we see in MDS that is to the inflammaging process linked to this. So thanks to the RNA-seq data that we have on the analysis, we can correlate those findings to the S189 and FTO pathway. So it seems that this pathway is important. We've actually produced S189 FTO mice, which we're in the process of characterizing. We're hoping that when they have both genes, transgene overexpressed, that these mice actually have an acceleration of disease progression. And we observed that there's splicing changes that occur at specific events. There's not all of the type of splicing that gets changed, but in particular, the alternative 5', SE and MXC are increased, and RI is decreased, for instance. So basically, there's these changes in spliceosomal changes, they lead to changes in the spliceosomal machinery itself. So it would be interesting for us to see, and that's something we're trying to understand, how is this changing over time. Hopefully by next time we have a meeting like this, we could present the data on the DRIP sequencing and the M6X sequencing. And we're also trying to understand, for instance, with human endogenous retroviruses, if that's one of the ways that RNA-DNA hybrids are happening in the cytoplasm. And with that, I'd like to acknowledge the work of our team, the Wei Lab and the Dr. Lee's lab, as well as John Cleveland, Eric Padron, and Kenneth Wright, who's giving us information on the human endogenous retrovirus. Thank you.
Questions? So it was not clear to me how the splicing changes you observed in your transgenic mouse replicated those you saw in MDS patient with S.E.W mutations?
Yeah, so DES-189, we have originally shown that replicates a lot of the morphological changes in MDS. In the bone marrow, it shows a lot of the dysplasia we've seen. Actually, a lot of the, even though I didn't put it, a lot of the information that we've seen in the, with FTO, when we tried, we've actually found it first on the mice, and then when we look at the human samples, it actually match exactly what we've been seeing. So that has been reported by us before, and so we have seen that. So based on the fact that FTO seems to be an important driver of this, what we wanted to know if it is, if it is something that you have to add as well for it to come faster. What we've seen in the mice is that you only see that accumulation of FTO after you age the mice, for instance, 12 months. That's when you start seeing a lot of these changes. So what we thought is like if we have it overexpressed from earlier, you can see that at an earlier time point because you're inducing a lot of these changes earlier on the mice.
Okay. Okay, thank you very much.
And our last speaker today, Mark Reinmakers, will speak about the niche, the hen or the egg. Thank you.
Thank you. Thanks to the organizers for inviting me. So what I will do is update you on the conceptual advance of the role of the niche in MDS. And then the organizers also gave me this title and question, hen or egg, that I will try to answer, or at least contribute to the answer in this talk. I do not have relevant disclosures for this talk. So, as elucidated in the previous talks, we know that genetic and epigenetic events are critical to the pathogenesis of MDS. So Ulrich's talk, Stein Erik's talk have recapitulated that. There is genetic events and that drives clonal evolution. At the same time, I think one of the questions that is still to be answered is why do all these mutations occur? And if they occur, why do they gain competitive advantage? And that's not so clear when you look at some of the common mutations like splice factor mutations or TET2 mutations that do not necessarily seem to have a competitive advantage intrinsically. So these are, I think, questions that need to be answered and that really asks for a comprehensive view on the bone marrow and MDS pathogenesis and the role of the microenvironment, which, at least in the postnatal bone, is the bone structure itself. Now, there is many niches, there is distinct niches, but for this talk I will mainly talk about mesenchymal niches that have been shown to be important for regulation of stem and progenitor cells. So this is data of some years ago now, where we showed that if you specifically change in a mouse model mesenchymal cells in the bone marrow, you can actually drive alterations in hematopoietic cells that are reminiscent of human MDS, and that actually gives rise to a clonal neoplasm, AML, that has non-random genetic abnormalities. So this really supports a model of, if you want, niche-driven leukemogenesis. Sorry that I'm having a stable view here, but you don't. So I wait for a second. Yeah, thank you. And this has been recapitulated in several mouse models now, that if you change the microenvironment, you can actually induce neoplastic disease. So this really raises questions, and I've summarized some of them and will address them briefly in this talk. So is there indeed a necessary contribution of the microenvironment to MDS initiation and evolution? If so, what are the molecular determinants of such contributions? And how then do I see that these alterations contribute to MDS pathogenesis? Now, what we in the lab do is we use these monogenic bone marrow failure and leukemia predisposition syndromes to shed lights on this. And that's basically because you can model a monogenic disorder. And one of these disorders is Shwachman-Diamond syndrome. As you know, a syndrome characterized by bone abnormalities and blood abnormalities, in particular neutropenia, but also a very distinct predisposition for acute myeloid leukemia. And what we do in mouse models is try to deconstruct the pathogenesis. So knock out this causative gene, SBDS, in distinct compartments in the bone marrow, either the mesenchymal environment, hematopoietic stem progenitor cells, or endothelial cells. And what we found when we specifically deleted SBDS in mesenchymal cells is summarized here. This leads to activation of p53, and then an inflammatory profile, including overexpression of the DAMP S100A8 and A9, that partially through TL4 signaling induces distinct abnormalities in stem and progenitor cells. Hyperpolarization of mitochondria, ROS, and genotoxic stress, DNA damage, which is accompanied by cell cycle checkpoint activation. So here is a link between a human disease relevant stromal abnormality and bone marrow failure. Now, is this relevant for human MDS? This is showing in a cohort of low-risk MDS patients in niche cells expression of S100A8 and A9, and you can appreciate that there is a subset of these patients that overexpress S100 in their niche, and that's confirmed here at the protein level, where you can see that some patients have quite high expression of S100 in these endosteal niche cells. And important to emphasize that these low-risk MDS patients with this niche inflammatory profile were not discernible using our standard prognostic parameters, IPSS, IPSS-R. Yet we did find that if you have this inflammation in the niche, you have a substantial higher chance of leukemic evolution at a much shorter interval. And that's then reflected in reduced EFS in these patients. Now, this really converged this data with data from Sheng Wei's lab and Alan List's lab, and also Ben Ebert's lab, that point at a common role in MDS, where S100A8A9 drives through the NLRP3 inflammasome, genotoxic damage, pyroptosis, and ROS. And it also indicates another, I think, relevant phenomenon, and that is that while S100A8, A9 may be everywhere in the bone marrow in MDS patients, the localization may actually determine its effect on a phenotype. And I mean that if you have overexpression of S100 in a macrophage or erythroid island or erythroid niche, this may contribute to anemia, as been shown by Rebecca Schneider in Ben's lab. And if you have overexpression in the mesenchymal stem cell niche, this may actually help drive genotoxicity and perhaps also clonal evolution, as I will show you later. Now, is S100 everything? Just a couple of slides to show you that that's clearly not the case. When we looked at the gene expression profile of niche cells, and these are cells that we directly isolated from the MDS marrow, so no expansion ex vivo, we show that they are distinct from normal bone marrow niche cells, and that this distinction is characterized by inflammation, among others. And here is just some of the inflammatory ligands that are highly overexpressed in MDS niche cells, and among them are some known negative regulators of hematopoiesis and erythropoiesis. Now, another interesting finding is if you look at this cohort, we did this in 45 low-risk MDS patients, that when you, if you compare the niche profile of patients that carry splice factor mutations and compare them to patients that have mutations in epigenetic regulators, that there is actually very common abnormality. So this inflammatory profile you can find regardless of the genetic phenotype. And I'll come back to that in my last slide. Now, what is the drivers of this inflammation? One of them may be that there is NF-kappa beta activation. That was suggested by transcription analysis, where we showed NF-kappa beta activation in these niche cells. One of the associated genes, NFKBIA, which is upregulated if NF-kappa beta is activated, was upregulated in many low-risk MDS patients in niche cells, and that's corroborated here by PP65 staining, where you can see that clearly NF-kappa beta is activated in hematopoietic elements in the MDS marrow, but also in these niche cells. And this is showing the same thing, but now by immunohistochemistry. Now, is this relevant? So when you overexpress NF-kappa beta in a relevant cell type, a mesenchymal cell, that's done here, overexpressing IKK, an upstream kinase regulator of NF-kappa beta, you activate NF-kappa beta in the stroma. What you see is indeed overexpression of many of these inflammatory regulators, and in co-culture systems, this results in reduced numbers of stem and progenitor cells and also reduced colonies. So NF-kappa beta may be an important common driver of this niche phenotype. Now, is it the chicken or the egg? The question that I had to answer here. I don't know. I can start saying that we have never been able to convince ourselves that there is mutations in niche cells in MDS. Doesn't mean that they're not there. We haven't found evidence for it. And what we did see initially when we compared these gene expression profiles of these cells that we directly take from the MDS marrow, and we compare them to expanded cells, niche cells from low-risk MDS patients. And here we used data from the elegant studies of Hint-Medjuv, that what you see in these primary cells directly in the marrow is not fully recapitulated ex vivo. So many of the inflammatory genes are upregulated only in the tissue. And when you culture these cells, they are lost. And that's again reflected when you compare then expanded cells with primary cells in these signatures that show inflammation. So this suggests that clues from the marrow are important for these inflammatory alterations. Now, in vivo support for this has come from studies in our institute from Rebecca Schneider. She uses the 5Q model of MDS, where she, in a hematopoietic specific manner, knocks out in a haploinsufficient manner these key 5Q genes. And what she observed is that then you see upregulation of S100, A8, A9 in these SCA-positive niche cells. This was also found in 5Q patients, and Rebecca confirmed what we also showed, that here in the 5Q marrow you see these niche cells that overexpress S100. Now, she went on to take cells from this mouse model, differentiate into macrophages, and then co-culture with mesenchymal cells, and then showed that by doing that you upregulate S100 in your mesenchymal cells. And similarly, when you overexpress S100 in these cells, this is conferred to the mesenchymal environment. Now, so in answer to this question, hen or egg, I think it depends on the clinical context. In a congenital bone marrow failure syndrome, like Noonan, like Schwachman-Diamond, you clearly have a hit in the mesenchymal cell that can drive these alterations. In MDS, I think it's different. It's more likely that there is a mutation in a hematopoietic cell that then changes the stroma in such a way that it starts contributing to disease pathogenesis. And also here there is ample of mouse models that show that hematopoietic cells can induce alterations in the stroma. Now then, coming towards the end, what does this all mean? How should we integrate this in a view of how the niche can contribute to clonal evolution and MDS pathogenesis? One slide, preliminary observations, is that going back to S100, what we observed is if you add S100 to wild-type cells, by genotoxicity, you actually get apoptosis and reduced CFUC. When we took DMT3 deficient cells, this was less so. These cells appear to be more resistant to this S100 stress. And similar things have been shown and are emerging, that some of the mutations that we often see in MDS actually carry some resistance to inflammatory stress. So what may this mean? And this is my last slide, and I have to say this, I think still, to at least some extent, is hypothetical. What may be the role of the environment in MDS? Well, maybe that you have these mutations that we all know. These mutations may have common downstream effects, and one of them may be innate immune signaling, that is enhanced. And again, there is emerging data on that. And there is a very interesting recent review from David Selman and Alan Listenblatt that highlight this. This may include NF-kappa beta activation, S100 expression. Now, this enhanced inflammatory profile signals to the stroma and may induce NF-kappa beta or Wnt signaling that I didn't talk about, or cellular senescence. These things are not mutually exclusive in the mesenchymal environment. And this leads to an inflammatory profile that creates a genotoxic environment. Now, depending on what niche we're talking about, this may affect different cells. But if you talk about the stem and progenitor cell niche, it may lead to functional repression, as we've shown in the example of S100. And this may lead or contribute to the bone marrow failure. At the same time, these mutated cells may be relatively resistant to that very same inflammatory stress. And that may contribute to their clonal selection in comparison to wild-type progenitors. And in addition, this genotoxic environment may or may not contribute to additional genetic events that ultimately drive progression, as was highlighted by Ulrich. So this is a model and needs more work. I think it's exciting because it would really indicate that targeting this signaling may ultimately not only have prognostic significance, but also help prevent evolution of these clones in MDS. Now with that, I'll stop with thanking many people, specifically the students that did the work, and Rebecca Schneider in our department, which we have a wonderful collaboration. Thank you. Happy to take questions.
Yes, please. Yes.
Oh, now it is. Mark, great talk. Thanks a lot. You know, I'll start with the question on your intriguing model. So one of the questions that I have in my mind is, you know, the transformation. If you look at DNMT3A or TET2 or mutated clones, they only develop an MDS, for instance, in less than 1% of cases. So if this is a general mechanism, so if these mutations really induce this inflammatory environment in the stroma, what in your mind, or if you could discuss this, why do you think it's such a low probability event if you know, the genotoxic environment is a general outcome of these early mutations? That's just something I can't stop thinking about, and I don't have a good answer, so would love to hear your thoughts.
Yeah, so that's a good question, and I think a difficult answer. So one of the things that I think we don't really know yet is that these things, eventually when we diagnose an MDS patient, it's everywhere in the marrow. And it may be, among others, that this inflammation is contagious, but it starts somewhere in a very local topic environment that we don't yet have a grip on. And so it may be that a very local event that we are, with the current technology, are not able to image or survey, that there it all starts locally. And it could be in a single cell, a TET2 mutation or whatever. And that is then the beginning of a spread that leads to a more global environment where these things are promoted. But I think it's, clearly work is needed to really define that, and that's going to be a tremendous challenge.
Okay, so if no further questions, thanks a lot. Mark? This is the end of the session. Thanks a lot for all the speakers and for you, and have a nice break. Thank you.